Today we are going to be talking about the cell cycle. So we're going to go over the different components of how a cell goes from being a single cell to dividing into multiple cells. So we're going to go through mitosis, which is where one cell just produces two identical daughter cells, and meiosis, where one cell produces 4 haploid cells, which are kind of like half cells. We'll get into that in a little bit. But first, we are going to start off with this circle-looking thing just to go over the overall cell cycle of division. So we're not talking about meiosis right now, we're just talking about mitosis, which is one cell into 2 cells, just dividing. So there are 2 parts of the cell cycle. There is interphase, which has the G1 phase, the S phase, and the G2 phase. Now, in the G1 and the G2 phase, it's just cell growth. It's getting ready to divide. It's making sure it has enough nutrients, it has enough material to actually split into 2 viable cells. Now, the S phase is where the genome replication occurs. So I'm going to go ahead and note down here that the interphase part of the cell cycle has the G phase, the S phase, G2 phase. So now over here, in purple is something called the M phase. And so the M phase is going to have something called mitosis and cytokinesis. Now within mitosis, there are 4 steps, and the 4 steps are prophase, metaphase, anaphase, and telophase. And so I'm going to go ahead and note down here that the parts of M phase are mitosis, and I remember this by the acronym PMET, for prophase, metaphase, anaphase, telophase, and then cytokinesis. And so cytokinesis is where that cell cycle kind of ends, and then once the cell divides, it follows back into G1, S, G2, all that. There's also a G0 phase, which is just nothing. It's not getting ready to divide, but that is not on our chart. Now we are going to move into the nitty-gritties of mitosis. So if we look over here, I already have the 4 stages labeled from what we said they were earlier. And mitosis, again, is just taking one cell and turning it into 2 identical daughter cells. So right now, the cell is considered diploid, which is also called 2N. The Ploidy, I never know how to say that word, but the cell is 2N. And so that's basically saying how much genetic material a cell has. And so since we are producing 2 identical daughter cells, it's going to start at 2N and it's going to end at 2N. And I know you might think during the S phase, it replicates its genome. So isn't that double the genetic material? So would it not be 4N? But you have to remember that the genes being duplicated are not providing any new information or new genes. So the Ploidy, I don't know, the Ploidy is still 2N. So we're not adding or taking away any new information. It goes from 2N to 2N, which is also called diploid. Now, haploid, on the other hand, is 1N, and we don't get into that until meiosis, so we'll do that in a second, but for now, just mitosis. We start off with prophase, and during prophase, this is kind of when the chromosomes start to condense, the DNA gets really close together, and we start to see those, what we think about when we think about chromosomes, those 2 X's. So they become visible, and then the nuclear envelope around it dissolves, because we're getting ready to separate out the DNA into 2 separate cells, so we need the nuclear envelope to kind of go away. So in the prophase, the biggest things that happen are chromosomes condense, nuclear envelope disappears, and then a spindle forms. I'll write that down. So we can see that our chromosomes are condensed. We can see those X formations. There's no longer a nuclear envelope, and the spindle that forms is going to be made out of microtubules. And these are going to be what attach to the center of the chromosomes at the kinetochrome, and it's going to eventually pull them apart. But the next phase is metaphase, and metaphase is where the chromosomes line up in one line straight down the cell. So as you can see, the chromosomes are straight down the middle of the cell, and these spindle fibers are fully attached to the chromosomes at this point. So they aren't being pulled apart yet. Prophase, starting to condense, spindle starting to form. Metaphase, they're lining up, the spindle is attached. Now, anaphase is when they start to get pulled apart to these separate poles of the cell. So, as you can see, the chromosomes are fully separated, and that little gray line in the middle is called the cleavage furrow. And this is just straight down the center of the cell, and that's going to help the cell split, and it's going to help the cytoplasm split. Now, telophase is where the chromosomes are completely on the other side. They start to decondense, they start to turn back into the normal type of genetic material that we're used to, and the nuclear envelope or membrane around the genetic material is going to start to form. So we're starting to form two separate nucleuses on either side of this one cell. Now, the cytoplasm is still intact here. As you can see, it's starting to pull apart, but the cytoplasm doesn't completely separate until cytokinesis, which, if you remember, was the second phase in our M phase. So let's go back to telophase. Telophase, like we said, nuclear membrane forms around each end, chromosomes decondense, nucleus is starting to become visible. That is mitosis. Now, before we get into meiosis, I want to kind of go over the difference between sister chromatids and homologous chromosomes. So sister chromatids are what form during DNA replication. It is an exact copy of the chromosome. So if we have chromosome A, for example, its sister chromatid is another chromosome A that was duplicated to be the exact same. So sister chromatids are just the exact same genetic material, the exact same makeup. They're literally just duplicates of each other, sister chromatids. And that is what gets separated during mitosis. Let's write that down really quick. So we talked about mitosis, we'll get to meiosis in a second, but also know that the sister chromatids are what separate in meiosis 2 as well. So mitosis and meiosis 2. Now, homologous chromosomes, on the other hand, these are what come one from each parent. So these are different. They're not identical. They have the same genes, but they don't have the same alleles. So they won't code for the same thing. So they're similar, but they're not identical. And so this leads to genetic diversity that we will see in meiosis when we go through in a second. And these separate during meiosis 1. So you only see homologous chromosomes in meiosis. You don't see them in mitosis, because in mitosis, we're not creating new genetic diversity, we're just splitting the cell in half. So let's write that down. All right, and I'm gonna draw arrows just to each one of these so we can remember. Sister chromatids is mitosis and meiosis 2. Homologous chromosomes is meiosis 1. Now, for some reason, this picture helps me remember sister chromatids, because sister, for some reason, I think of 2, and I have 2 arrows, one to meiosis, one to meiosis 2, one to mitosis. So that kind of helps me memorize which one goes to where. But now that we know the difference between those, we can kind of go into the nitty-gritties of meiosis. So starting with meiosis 1, this results in 2 haploid cells. And these cells each have a copy of each homologous pair. And this will make sense here in a second once we go through each one of these phases. Now, the 4 steps are the same as mitosis. Meiosis 1, meiosis 2 are both prophase, metaphase, anaphase, telophase. Exact same. Only thing that's different is the type of genetic material that is being separated. So we'll go through it slowly. Prophase 1. This is where the 2 sister chromatids condense and they start to form homologous chromosomes. And so this happens during crossing over. And so crossing over, this is just important to note that crossing over occurs in prophase 1. That is important to know. Crossing over is basically when genetic material from one parent and the other parent switch. So if no crossing over happened, that means that all of the gametes from meiosis 1, meiosis 2 will have all of the genetic information from either the mom or the dad. But we know that just from experience, that's not how genes work. You don't get all of your traits from one parent. There has to be some sort of mix and match. And that happens during crossing over. This is where genetic diversity happens. Prophase 1, genetic diversity. Okay, and we will jot all that down. Again, like we said, this time we can see the 2 pairs of sister chromatids forming homologous chromosomes, and then we can see the crossing over, and then the spindle again forms because it is prophase. Next, metaphase. This is where everything lines up at the center, just like mitosis, the exact same thing. They are all lined up. The homologous chromosomes are lined up. It is important to note that. Next, anaphase. separating out. Spindle is there, they're going to different poles, same thing. Remember, the thing that is separating here is the homologous chromosomes, and as I noted, these sister chromatids are remaining together. And so if we zoom into our picture, we can also kind of see how the blue and the pink are a little bit mismatched. That is a result of crossing over. That's what I mean by we're switching genetic material, we're playing mix and match. Lastly, telophase. Exact same thing. The material decondenses and envelope forms, and we start to see the nucleus. Okay, and that is it for meiosis 1. So we finished meiosis 1. Now we have 2 daughter cells that are haploid. So they only have half of the genetic information that we started with. And this is because it was the homologous chromosomes that separated and not the sister chromatids. If the sister chromatids were to have separated, we would still be diploid because it's the same material getting separated, so we're not losing any genetic information. But because it was the homologous chromosomes that separated, we went from diploid to haploid. So we did lose some genetic information. We only have half of the information now. Moving on to meiosis 2. Meiosis 2 is where we finally result in 4 haploid daughter cells. We are going into meiosis 2 as haploid, and we are going out of meiosis 2 still as haploid. So that should give you a hint to tell you that it is the sister chromatids that are separating because, again, being redundant, but sister chromatids are the exact same copies of one another. When they separate, we're not losing any genetic information. We can kind of go through this one a little bit faster. Prophase, we've been through this. Sister chromatids condense. New spindle forms, nuclear envelope disappears, same as meiosis 1, same as mitosis. Only thing to note, remember, sister chromatids. Metaphase, remember metaphase, they're lining up, metaphase plate. All right, sister chromatids line up. Anaphase, getting separated, same thing. Sister chromatids getting separated this time, same as mitosis. And lastly, telophase, decondense, envelope forms, nucleus starts to form on either side. Okay, let's go ahead and note if each start and end product of each of these is either diploid or haploid. So mitosis, it goes from 2N to 2N. Meiosis, remember we go from 2N diploid to 1N haploid because the homologous chromosomes separated. Meiosis 2, 1N, because that's where we ended from meiosis 1, stays at 1N because homologous chromosomes are not separating. It is the sister chromosomes that are separating. So lastly, before we end this notes page, I just want to go ahead and write down in bullet points the critical differences between mitosis and meiosis. So let's write it down here. Mitosis versus meiosis. Start with mitosis. Products, 2 daughter cells. Sister chromatids separate. Remember, identical. No genetic variation here. Now let's go to meiosis 1. Meiosis 1, the chromosome number decreases by 1/2. Now, remember, homologous chromosomes separate. We only have half of the genetic information that we started with. Results in 2 haploid cells, which is 1N. This is where crossing over occurs, which is the exchange of genetic material, which leads to genetic diversity. And here, the sister chromatids, they remain attached, so they're still together. Lastly, let's do meiosis 2. This one is similar to mitosis. The only difference is it's haploid, where mitosis is diploid. Here, sister chromatids separate, and it results in 4 genetically diverse haploid gametes. So all of these lead to genetic diversity. So meiosis, genetic diversity. Hopefully this is all coming together and this all kind of makes sense. It'll take a little bit of practice, but this is a good overview of what's going on. The most important things to remember, I would say, are which ones lead to genetic diversity, the ploidy of the cells, which stages do the sister chromatids separate versus the homologous chromosomes separate, when does crossing over occur, prophase 1 of meiosis 1, and then that is pretty much it. We didn't really talk about cytokinesis. The only thing you really need to know about cytokinesis is this is when the cytoplasm actually splits. So I'll just write a note real quick. And this is due to actin, myosin, which is a part of muscle, which we will talk about at a later date. Actin and myosin wrap around the cell and they pinch to separate them using ATP. So I'll say actin and myosin pinch, use ATP to do that. Okay, all right, so that really is everything that I have for you for the cell cycle. All of the notes will be on the link tree. It will be under biology and then cell cycle. And that's all I have for you today.